Again, the structural proteins found in your coronavirus are the following. Spike glycoproteins or S proteins that are surrounding the virus, M protein, N protein, and E protein. So your S proteins are cleaved into S1 and S2 subunits. The S1, take note, is the one that attaches to the ACE2 receptor, which enables the virus to move into the cell. So once it enters, it opens up, releasing its mRNA into the cell. This mRNA is a positive sense single-stranded mRNA, which means that it is ready to read right away to the host ribosome. When the mRNA goes through the host ribosome, this process is called translation. Translation gives us protein as this mRNA is being read. This creates two polyproteins which are now processed by the main protease through the process of proteolysis that cuts them into many more smaller proteins and assembles them to what we call replication and transcription complex, or RTC. This RTC is now an important machinery to help create more coronaviruses. RTC is used in different ways. One of the ways it is used is to make more copies of this viral genome. It's used to run those mRNAs through, and as a result, it produces an identical copy. Except that the identical copy is at the opposite direction, a negative antisense single-stranded mRNA, which needs to get back to the positive sense. So this negative sense needs to get back to a positive sense in order to build new viruses. This negative antisense runs back through the RTC, which produces again a positive mRNA. These processes are called replication because we're taking a full strand of mRNA and making its complementary strand. This replication of the viral genome will require RNA-dependent RNA polymerase enzymes, or your RDRP. Another way of using RTC is again running the positive mRNA through what is now called your discontinuous viral transcription, which creates some smaller fragments. Again, opposite directionality, so it's negative, compared to the replication process, which creates a continuous genome. These smaller fragments are called subgenomic mRNA, which codes for the four structural proteins, your S, M, N, and E proteins. Again, this is negative, so it needs to go back to its positive form to be read by the ribosome. The subgenomic mRNA runs through again the RTC, which is called transcription, a discontinuous transcription, which reverses its direction, which now makes it positive. This can now go through the ribosome, which, as mentioned a while ago, can produce a protein. So, let's say for example, this bit of spike protein mRNA code that goes through the ribosome produces proteins. Again, this is called your translation, which produces proteins, your spike proteins, which eventually builds up. The same goes for the other structural proteins. So all of these proteins help create new viruses. Other small subgenomic mRNAs coding form S, M, and E proteins that are translated at the ribosomes of the rough endoplasmic reticulum, or RER, to enter the endoplasmic reticulum, or ER. The ER packages these proteins and transports them to the Golgi intermediate compartments, or your ERGIC. So we now have structural proteins from transcription, fresh copies of mRNA from replication, and nucleocapsids in the cytoplasm. So viral nucleocapsids in the cytoplasm bind to the ERGICs and are pulled in, forming the viral envelope embedded with S, M, and E proteins. The intact viruses are then packaged in smooth secretion vesicles by the Golgi. The intact viruses are now released from the host cell by exocytosis.